Yay, okay, I think it's working.
I kind of want to do this in my hair.
Okay, we're at gymnastics right now. So let's see, waiting to see if you guys, what do you want me to do with this here? Right, okay. To see if you guys got the link. Let's see. I'm trying to see how to share it just to make sure. Okay, here we go. Okay, now I'll go back. Let's see if I can share it for everybody. Okay. Go back and fix this. Okay. I'm gonna try to get started here in a minute. Okay, let me get back to this. Okay. All right. Except Felicia, I think she's planning to join. Is able to go through it again later. All right. All right. Okay. Let's see. I think it's just me for now. And 4:05. So let's see if we can jump into chapter 8. Get my binder. I'm supposed to be here at 3:45. And just running late today. So not as prepared as I was planning to be. Okay, let's see. Alright. Turn on my sound so I can hear if anybody comments. And okay, let's see. Okay. Okay, well, I guess I'll just check back on that in a minute and see if anybody else needed help getting set up with that. So I'm going to try to stay on chapters. I don't know how interrupted we will get being over here at gymnastics. So I did make these study sheets that are on Dropbox that you should be able to use. Alright, so chapter 8 was on bacterial genetics. And so she was just talking about like the genotype and how changes are made to the genotype of organisms, bacteria, so bacterial organisms. So that's there at the top if you have your study sheet. So I guess maybe we'll just walk through the chapter and I'll walk through the chapter of the book as well and see if that's helpful for anybody. Okay, don't know where to start. I wish somebody was helping ask me questions to get me started on this. So we'll just go through her notes. So she started off talking about a mutation that gives it a huge advantage. Then that's part of natural selection where, it's able to just jump out into the environment and kind of do its thing. So it favors those with greater fitness. So we talked about the difference between genotype and phenotype. So genotype refers to your genetic makeup, and phenotype is going to be what you look like. Okay. So then we talked about genetic change in organisms. She jumps into that. And so you're just going to compare mutants to wild type. So everything kind of goes back to wild type whenever you're looking and making those comparisons. Okay, so the way that things can mutate, there's a few different, so she did talk about mutation rate. So that's between two different organisms to see how similar they are, or, for like HIV when they started making changes. She talked about horizontal versus vertical gene transfer. So in this, that's where we have, so this would be horizontal gene transfer, where you have bacteria trading genes horizontally, like to other living organisms. And then you have mutations is another way that you can have sort of gene changes. Then any of these changes, like for a mutant, a lot of times the mutant will just die. But if it's able to, where, you'll grow it in something that contains tryptophan, and then you'll do different experiments based on that. But if it survives these things, for example, this is usually a beneficial thing, then it will pass it on, and that becomes vertical gene transfer. Reversion is when you have a mutation that goes back to its normal state. Point mutation is the change that's one of the most common changes that is a spontaneous mutation of bacterial, that's bacterial genetic. It shows you how you have a few different mutations. So it shows you the outcome based on what changes and how it affects the protein, the amino acid that it's going to use whenever the ribosomes are looking at this. So in a point mutation, you're just taking one thing and you're just changing one of those bases. And then it can result in a silent mutation where it ends up making the same amino acid anyway, or it can be a missense mutation where it's a different one, but mutation where it codes for a stop codon and then it just cuts off the protein and right there. And it's typically, almost all cases, I guess, it's going to. Anybody else needed help? I guess I would see it pop up on my phone. Okay. Look at all that stuff on there. Okay. So we'll keep going. A mutation that inactivates a gene is null or knockout. And so when people are studying bacteria, they will do that on purpose sometimes to knock out a gene if they want to study a certain thing. So another mutation that you can have is a frame shift mutation or removed. And so that's over here, frame shift. And a lot of times it will end up soon after that causing a stop. So that was when she wrote out the cat ate the rat. That's, if you stop reading those 3-letter words in the correct frame, if you're not using the right reading frame, then it's going to not make any sense. And that's why it's a nonsense. It ends up being a nonsense mutation with base substitution. And for frame shift, it's going to be a knockout because typically it's going to cause an offset that she talks about transposons, which are jumping genes, and these all fall under spontaneous mutations. So that's another thing that can just happen. I still don't really understand transposons very well, but I know that they're just groups of genes that can jump around and that they have to have an insertion sequence to be able to pull from one part and go into another part of the gene, and it will cause insertional inactivation. So if that gene comes from one place and jumps into another place, it messes it up. It stops it from functioning properly. So in that process is called transposition. And she talked about corn. Okay, so then the next thing is going to be induced mutations, and so it's 4:15. So we'll try to keep going through here. When you're looking at induced mutations, these are things that are being caused by outside sources. There's a nice chart in the book. You can tell that I like charts. And that's right here where it's showing a chemical mutagen. And then on the next page, it's getting into the other mutations and kind of talking about what happens and the result. So what is actually being caused by that mutation, and then what is the result of it. Okay, so the types of mutations that we can have that are induced mutations are chemical mutagens, transposons, and radiation. And those have other like subunits inside of them. So for chemical mutation, that's a chemical that's it can cause base substitution. So we learned about that. That's a spontaneous mutation. But if it's caused by a chemical, it's going to be an induced mutation, but it can cause a base substitution. Or it can cause a frame shift mutation. So, it just kind of messes up those bases and causes those other things. So the types of chemical mutagens you can have are base analogs, which is something that's, like an analogy is something that's like making a comparison. So a base analog, so an analog is similar to a base, but it's going to have different bonding properties. So it's not the same as having the correct base there, and it's going to cause the molecule to not function correctly. The other thing that you could have is called, I don't know how to pronounce it, but intercalating agents, where it gets in between the molecules of DNA. Chemical mutagens, then transposons, I don't know how, but there's a way to force transposons to inactivate genes. That can be a type of induced mutation, as far as I understand it. The other type of induced mutation is radiation, and we looked at UV and we looked at X-rays. And UV is going to cause thymine dimers. So if you have two thymine molecules in your DNA. The other thing is X-rays, which cause single and double strand breaks in the DNA. And sometimes they can come back together, but sometimes they kind of like don't line up the right way. And that happens. Okay, so X-rays, let's see what else we have. Okay, the next thing that she gets into is DNA repair. And you can probably guess that, so all this is in the book. So we got thymine dimer of damaged DNA. So these are the different ways that you can have repair of damaged DNA. And I did add that on here. So if you have spontaneous mutations, then initially you're going to have proofreading by DNA polymerase, which is very efficient at proofreading. And the other thing that you can have is mismatch repair. Where a protein binds to the site of the mismatch and just cuts it, and then they break down the DNA and rebuilds it, and hopefully it's right that time. In the book, it says gives another one, but I don't know that she mentioned that. The next one is for mutagen-induced things. So this would be induced mutations. So I guess that actually is wrong on my paper, right? Because it says mutagens, and that should really be mutations. Because a mutagen is going to cause, I just named these, it's something that is going to induce a mutation. It's something that's going to cause a mutation. So I need to go back and fix that on my paper whenever I go back and look at it. So hopefully I'll find some more things like that we can learn about as we go. Then if you have repair, you also have, proofreading for induced mutations. You kind of have the same thing. You can also have fix that. Photoreactivation, which uses light to use some different enzymes and stuff. I think the energy of light is used to break the covalent bond between the thymine dimers and make them straight again. Or you can have excision repair, which can be done in the dark, but it's not as good. An enzyme is going to go in and remove some of the DNA. So excision, you're thinking like you're cutting something out. So it goes in and kind of cuts out the thymine dimers and then fixes them. But there's more opportunity for error, I guess. So it's like preferable for it to just use light repair when it's going to just, you have the same two thymines and they get stuck together and you just fix them. So that's probably the best thing that it would prefer to do. And then SOS is like the last option that it has. If nothing else has worked, it just kind of goes in and chops up all kinds of stuff and hopes that some of it is fixed enough for it to not die. But it still will be mutated after. That's when you're looking at selecting mutants. So I just put like some vocabulary words up here at the top, just like threw them in because I didn't feel like it fit in the organization. So where I put auxotroph and prototroph, so a lot of that is like on the Quizlet, you can go and look those things up. Reversion and the Ames test, whenever they use a mutant, whenever it back mutates, then it's a carcinogen because it caused a mutation. And okay, so direct and indirect selection. So direct selection is if you have, let me make sure I'm getting this right because it is confusing for me. So I think direct selection is you are growing something on a plate that will grow antibiotics. Then you would grow it on a plate that has, for example, like streptomycin. So you have streptomycin and you grow the stuff on there. If you have a streptomycin resistant mutation or mutant on your plate, obviously it's going to kill the parents that are not mutated to resist streptomycin. So that's how you can get those mutants. And that one's pretty straightforward. The other one is a little trickier, which is called indirect selection. And when I first read those a bunch of times, it was like reading the same exact thing. It was very, okay, I don't know what this is telling me. So let me look at it in the book to make sure that I'm explaining it better. But it uses something called replica plating. It's a clever method for indirect selection. It developed in the 50s. So you use velvet. So you take your original colony that will have mutants and non-mutants, wild type cells, I guess it would be. So you put velvet on it, and then you put it in on another plate. And there's a chart in the book, and there's pictures. So then when those colonies start to grow, so I think you have two different plates that you're going to grow it on, and one is just glucose salts, and one is, what's the other one? Okay, one is nutrient agar. So one has all the nutrients in it, and one, yay, did somebody just join? See a person. Send me a comment if you can. I don't see where you are. If you can comment, go ahead and comment. I don't know. I just clicked a bad button. Okay, restore chat. Yay! Okay, a person. Okay, and you should be able to go back and read everything else. I'm sure this is like super boring, but if you have questions as we're going through the chapters, so yay! I'm glad you're here. I'm gonna type back and be like, okay. But I'm not gonna like type because I'm talking. So if you have questions, feel free to share questions, and if you have to go, just feel free to leave. I will not be offended. Okay, so I was talking as the glucose salts. Let's see, what about the glucose salts? It's so confusing. I get mixed up with this. Okay, the auxotrophs do not grow. And so on the nutrient agar, everything grows. So obviously everything is, okay, that's my one little mutant in here. So that's how you can identify an auxotrophic mutant through indirect selection, which is tricky. And if you didn't see this earlier, since you just got here, I did put up these study sheets. If that would help anybody. Okay, so we just have a few more minutes to get through chapter 8. So let me see what else I need to get through. So we talked about mutant cells first. So there's a few different ways that can happen. In the last couple minutes, we'll get through this. So you can have DNA mediated, which means that, so basically like if a cell dies, then, and it like bursts for some reason, the DNA just kind of leaks out everywhere. And if you have a cell that's competent and it's just like walking through, it's like, ooh, look at all this DNA. I will take this DNA and see if it's useful for me. So it picks it up. So that's DNA mediated, but you have to have a competent cell. And the next thing is transduction. And that's when you have that viral process go wrong, where the bacteriophage, instead of just having phage DNA inside of it, it has DNA from another bacterial cell. So that's transduction, and that is called a transducing particle if it's that little bacteriophage looking. Okay, so competence, we've talked about that. Conjugation is cell-to-cell contact, where, and she did have that in her slides, of the cells transferring their stuff over to each other. Okay, so then they do have stuff in the book for DNA mediated. You can go and look at that chart. Okay, I think it's pretty much getting close to the end of our time. Let's see what else is in here. Feel free to type questions if you have questions from chapter 8. That's almost it. Okay, so sometimes the chromosome can get transferred. It's for the F plasmid because that's a fertility plasmid. Okay, understanding that. So if anybody has anything to add about conjugation with bacterial cells, I know it's F. It's either positive or negative for fertility plasmid, including the F pilus or pilus. Okay, and then HFR cells stands for high frequency of replications of recombination. I had replication. Okay, high frequency of recombination cells. When the F fertility plasmid integrates into a cell, but it goes into the chromosome instead of becoming a plasmid. And it can be reversed chromosome, and that's called an F prime cell, which they talk about in the next section. If the plasmid comes out of the chromosome and goes and becomes its own little plasmid. But if there's an error, it's called F prime, and I don't know what that means. F prime, the chromosome. Oh, it leaves a chromosomal fragment. Okay, a piece of the bacterial chromosome is removed. Okay, if anybody has anything to add to that, that would be fine. Sometimes somebody else is here, say hi. Just talking through my notes is so fun. Sometimes it acts like terrible because it doesn't have time to transfer the whole thing. Then the last little thing, we're past our 4:30 mark for chapter 8. She talks about the core genome and the mobile gene pool, and that's just like, if you're looking at E. coli, 75% of it is all the same for all these different E. coli individuals, but this is the core genome, and these are the things that can move around and can give it extra genes or different genes. So some will cause diseases, some, are fine and are happy bacteria. We already kind of talked about plasmids. Low copy number versus high copy number was important because when we get into biotechnology, you want to use a high copy number, like if you're making insulin, and then resistance plasmids are things that they can trade to get resistance. Genomic islands, and then CRISPR is really cool where they can sort of memorize and keep track of those little viral particles from the bacteriophages. I did need to change this if you didn't see this. I need to change this to mutations because it's not mutagens. Mutagens are only for induced. So if you're here, you can say hi. Do you have questions, comments? Anything to add? This would be better with Steady Betty. I hope Hannah's feeling better. Hope it's not the flu or anything worse. Okay, so chapter 9. Let's see what that email was. Okay, it's just YouTube. All right, so getting into chapter 9 was talking about biotechnology and bioengineering, taking advantage of any of these bacterial processes. Yay, thank you. I feel like this is so boring without me talking with somebody else. Please ask questions if you think of something. or if you have anything to add. I don't know. I hope this is helpful. It would be better for me to have somebody to interact with, for sure. I'm not nearly prepared enough to just like feel like I'm trying to teach this stuff. So, okay, so chapter 9. So genetic engineering is when we're taking advantage of these little bacterial processes. You know, it's like little bacterial or factories that are doing all these things. We're like, oh, let's make the bacteria make insulin for us. You know, so that's kind of what biotechnology is about. And this is my little paper that I have up on the group that kind of overviews. So the kind of overview of the chapter is tools, and then kind of 4 different categories of biotechnology. So these are the tools we use for biotechnology. And then these are the 4 different things that, you know, kind of processes or procedures that we use. And it's modifying genes of living things. Okay, so to discuss the tools part, so we just looked at restriction modification systems, and that bacteria, that's like their primitive immune system that they use to, you know, get rid of these bacterial viruses or, you know, kill them before they take over the cell. So we take advantage of that to do some different things with biotechnology to just manipulate bacterial DNA or human DNA or proteins or anything that we want to do with that. And so part of the restriction modification system is restriction enzymes. And so restriction enzymes look for like a little segment of DNA, and it's typically like, I think is it a palindrome where it like goes back and forth. So it would be like scissors. And so it's going to make like an uneven, you know, so if this is your DNA, it's going to cut it like, you know, here and here kind of thing. So it's not going to cut it like straight. It's going to cut it in those little ends that overhang. How can I make it overhang? Okay, so like an end that overhangs. So this little piece that was cut is now a sticky end. And so it's going to, you know, and they're all cut the same exact way. So they're going to meet up together the same ways. So if you take bacterial DNA like plasmids, and then you take like human DNA and you cut them with the same restriction enzymes, then you mix them all together, they're going to all combine back together in different ways because those sticky ends will all meet up together. So what they look for, that's called a recognition sequence. And the little pieces that they make are called restriction fragments. When you mix it all together, so I didn't know that okay, genetic engineering, blah, blah, blah. Okay, well, I don't know. I don't see where it talks about cloning right here. But maybe that's a question that I need to flag if I have more stuff that I need to look up before Monday. Okay. So the next tool that we use is DNA gel electrophoresis, which I have no idea who came up with this, but it's just so interesting that somebody thought like, hey, let's take like some gel and put it on an incline and then put some little like, you know, jar things at the bottom, like little test tubes, and fill it up with this DNA that we made that they might took the DNA out of those cells. We like wouldn't see anything because there's not enough stuff there. So they make a lot more DNA. They break it all up with those restriction enzymes, and then they put it in these little, you know, tubes. And then they have it on, like the tubes are at the bottom of the thing, and then they run electricity through it. So DNA is negatively charged. I think that's right. And so basically, like as it moves through, you'll have the heavier, bigger, longer fragments kind of moving slower being at the bottom, and you have the little tiny fragments go to the top. And so like you can use it to sort the DNA based on where those restriction enzymes cut it, which is just really crazy to think about. But somebody came up with that, and they use it for all kinds of things. So it separates DNA fragments by size. We'll be able to come back together with anything that's been cut, you know, so you have like tons of places in the DNA where it's going to be cut, where you're going to have these sequences. And remember that came from the bacterial's primitive immune system. Then the other thing that tools they use in biotechnology is CRISPR, and they're trying to use that for genetic engineering, which is really, really exciting. I'm not sure that they get into a whole lot of stuff. Okay, yeah, I think there are some things that they do use it for now that are not just experimental. Yeah, so they can use it to attach molecules. They can use it. So DNA cloning involves isolating DNA from one organism, cutting it with a restriction enzyme, then transferring the DNA, a fragment of that DNA into a different organism. Okay, so whenever you do that, you make a recombinant molecule. So that process is actually called cloning. So like, oh, okay, I didn't know that that was what cloning was. And the DNA molecule used as a carrier of the clone DNA. So if you have a plasmid that's carrying, a little tiny piece of human DNA, like when they were mapping out the human genome, it would be called a vector. So it's usually a plasmid. Okay, and the DNA part of it is called an insert. So you have an insert that goes into a vector. And she did. It's fine. It's totally fine. Yeah, that's my friend. Say hi. Hi, friend. No, it's fine. I didn't have time to drive to the coffee shop, and I didn't have table space to put all my stuff out, so it worked out. Okay, so gene fusion, and this is something she said that they don't use anymore, where they would put a reporter gene into something. That was one of the things that she did put on the quiz. So they can use cloning for protein production. So that was insulin when she talks about insulin. And they used that to do the human DNA, where they organized all the Human Genome Project. Let's see, and then more than one organism. Like when she says, like, they just took a bunch of seawater and they just looked at all of the genetic information that was in there. That's called shotgun cloning, and it's to look at the metagenomics of a group of organisms. Okay, so then when you're trying to research something, like her example was, if you're trying to make insulin, how do you know that your E. coli are making insulin? And so that's why you attach a reporter gene, so that you know that if that E. coli is making insulin, it will glow green, because your reporter gene is fluorescent. Okay, so that was the end of that. That was the end of talking about genetic engineering in bacteria. So I think we talked about all these things. So, this is when you think of like GMO foods or, GMO animals. They're called transgenic. And they have introns. So that's another thing that you have to keep in mind is introns. When you're putting something into bacteria, and you also have to be aware of their metabolism. Okay, and when you put, I feel like I'm getting this here, did we not get into one little E. coli molecule? Or if you want to study, or I don't know, maybe it would, but if you want to study different pieces of the chromosomes, then when you cut it up, okay, so then if you have all these plasmids with human DNA, like this one has a tiny little piece of a chromosome, and this one has a tiny piece of a chromosome. When you look at all of that together, if you look at this whole big pile of E. coli plasmids, not E. coli plasmids, just plasmids. If you look at all these plasmids, you would then put them into E. coli, but that would be the DNA library of the whole entire human genome, if you wanted to go study certain parts. That's my understanding. The other thing, so when we're talking about introns, we're comparing organisms. So, when she's saying, like, when we get into later chapters and we're talking about how organisms are related, that it's really confusing. And it's especially confusing now that we can look at the DNA of the different organisms. And so what we look at is ribosomal DNA, which we'll get into that. I don't know when we get into that if it's in this chapter, but when you're comparing it, you're like, oh gosh, these organisms that we thought, we thought it was fungi, but they're not fungi now that we looked at it. So that's called genomics, and bioinformatics is when you're analyzing all this data from different genetic information body and looking at their role in health and disease. So that's DNA sequencing. Those are the two things that we use DNA sequencing for. So sequencing is like what order are the DNA molecules in. And then the next thing is PCR. which is polymerase chain reaction, where, like I mentioned earlier, if you have, if you're doing like DNA gel electrophoresis and you can't just use one tiny drop of blood, you couldn't even probably use a lot of blood. You have to pick your DNA, like get your little DNA sample, and then you're going to use the PCR technique to make 1 billion copies of all these DNA molecules. So then when you put it, you can actually see it when you stain it with the fluorescent dye. And that only takes a few hours. So before they had to use E. coli to do these types of things, and it took like a week to amplify all this DNA and make the E. coli amplify the DNA. But now that we have PCR, we can kind of use the little bacterial technologies to make these things happen. So let's look at some terms that she got into. So it's a 3-step cycle. Remember that we use enzymes, we use polymerase, because polymerase, remember, goes and builds the little DNA molecules. So we're going to use polymerase from TAQ. TAQ, Taq polymerase from Thermus aquaticus, because that's one of those hyperthermophile bacteria, and so their polymerases work at the temperature that DNA will melt. So when you have the polymerase in there, it can keep working, and you don't have to, it just cuts out a whole lot of steps. So you heat it up, denature the DNA, then you lower the temperature to allow the primers to anneal. So that's primers, where you're going to put primers in. So that's how you choose what sequences you want to copy. So you choose your primers. Like, that's something that the people do. They're like, okay, we're going to use this primer. Then you throw the primers in. They anneal, and that tells the DNA polymerase where to pick up what it's doing. Then you raise the temperature, and it's going to replicate the DNA. And it doubles each cycle that you're going to do this. It's going to double the DNA. Because if your polymerase wasn't heat functional, you would have to keep adding more DNA polymerase every time you would break up your DNA. Okay. So it's exponential. They typically use 30 cycles. And there's 2 types, which I don't really know what the differences are between regular PCR and RT-PCR, which is reverse transcriptase PCR, where it makes complementary DNA. So you take mRNA and you use it to make complementary DNA. So I don't know, so maybe that's only for things that you're having to use with bacteria. PCR, the reverse transcriptase, is when you need to have stuff that is compatible with bacteria. And then qPCR is where you have a fluorescent marker, so you can see how much you're making of your product, which I guess that's similar to using a reporter gene, which is what they used to have to do when they were using E. coli to do all these things. Okay, we look pretty good on time. The next thing is DNA probes. So if you want to identify a certain sequence of bacteria in your DNA, a certain sequence in your DNA, then you will use so you're looking for a specific nucleotide sequence. That's how they use like a rapid strep test or pap smears, was what she explained. Okay, and then, so then there's 3 techniques that you can use with these probes. Blotting is when, let's see, I haven't been looking at my book along with it. She has a picture in her slideshow of it. Okay, so I know that's right under FISH, but this is colony blotting. So you're going to take your colony and you're going to put it on a little nylon piece of fabric or, plate kind of thing. And then you flip it over and then attach. So you're like, oh, okay, these 2. And then you have to mirror image it when you go back to see. And that's because you wouldn't just want to break these down and then put your probes on it, because if you did that, you wouldn't still have your bacteria alive anymore to be able to study them. And so this is a way to find out what's in the DNA of your colonies without killing them. And then FISH uses I guess you can just call it fish, uses fluorescent probes with intact cells. So I don't know, what the difference is for that or what we use that for, but you can look at multiple things. With fish, you can look at multiple things and they're different colors. So you use a fluorescent probe. Well, I guess the other one is fluorescent too when you're using colony blotting. But with this, you can look at multiple things. And then the last thing that she talked about was DNA microarrays or biochips. And those were really expensive chips where you can expose it to something and then go back and test the DNA. Well, not test the DNA. You can just look at the colors of what it's doing. And those are super, super cool. So they can study different things. She used the example of coral. But later down the line, hopefully we could use it for studying people, like their responses to medicines, or I think that was the main example that like your dose and your type of medication. Okay, so that's pretty much the end of chapter 9. If y'all have any questions or comments about that, continue to listen to her lectures, continue to review your Quizlets. And the other thing that I have found really useful is the summaries in the back of the chapter. So that's, just really nice to go back and kind of read over everything all together in one place. Okay, so that's chapter 9. That's on chapter 9. Drink a sip of water. All right, so chapter 10 is talking about identifying and classifying microorganisms. This is my organization sheet that I'm naming organisms, which is important because when you look at chapters, I guess it's 11. I need to put a sign on the door like, you're going to be on TV.
I don't charge for mine.
Oh, okay. Awesome. Okay, so chapter 10, and then when we get into chapter 11 for that, those next couple of chapters. Okay. So she started off by saying that back in the day, we just looked at what something looked like, and we said, oh, hey, this looks like this, and this looks like this. Then the next thing, they started to look at physiology. So how does it function? They started to look more at function. Then they started looking at things like metabolism and just trying to get an organizational system for organizing these different microorganisms, especially tricky. Okay, so today, and that's why she did not want to use the book, is because today we are looking most specific, although you can, so species is a group of closely related isolates or strains. So you can have different strains in the same species. So like you could have different strains of E. coli, but in general, a species, because bacteria don't have sexual reproduction, she explained that, we can't look at, organisms that can. Taxonomy also includes informal groupings, which, like when I get to those other chapters later on, I do have informal groupings on those as well. Phylogeny is evolutionary relatedness, so that's looking at evolution and saying that how are these things related based on evolution. If something started here and then it branches out, then you have these two separate organisms for, way down the line. And she said that bacteria and archaea split a really long time ago, and then so eukaryotes are actually more closely related to archaea than they are to bacteria because that split was down here, and then we were farther down the archaea train. So now we have a 3-domain system that replaced the 5-kingdom system. She talks about Bergey's Manual of Systematic Bacteriology, where you can look up any organism and its characteristics. Getting into phenotypic identification, we're looking at, how does it stain? I think I have all those things in here. Okay, so for microscopic morphology, what is its size? What is its shape? What does it stain when you're using a microscope? Then, kind of bigger than that, you're going to look at culture characteristics. And you can even use different cultures like selective or differential media to be able to, see what grows in your culture. You can use metabolic capabilities, which is biochemical testing, which a lot of that I think is pH. And then you can make those keys where you say, okay, is it gram positive or gram negative? And then you're like, is, catalase, coagulase, and, you can identify things that way. And you can even purchase those as a kit where you do them all at the same time, and you can just figure it out super fast and not have to wait. So microscopic morphology is the first one. It's really fast. Some things are very easy to organize that way. Pheno is all under phenotypic characteristics. Okay, culture characteristics, differential media. And she gave examples of all of these, so you might want to be familiar with some of those examples. And then metabolic capabilities, you're looking at, what color does it turn this indicator? But it's my understanding of serology. Okay, so it uses antibodies for detection. So you're using antibodies to detect different characteristics of the organism, I believe, such as, okay, so it's using antibodies to detect specific proteins and polysaccharides. So you're using the antibodies to see surface structures such as cell wall, capsule, flagella, and pili. And sometimes those are characteristic enough to be able to detect it. And it says in the book that that's what they use for a quick strep test. Interesting. And then the next thing is looks for a protein profile in the organism. And what I understand this does is basically vaporizes it with a laser. And then as the ions pass out of the tube, it takes readings of different sizes of ions that are released. So it's really complicated. It's really complex. But I think it is vaporized. Leaves the molecules intact. So then it goes through this little machine and sees, and then it makes like a fingerprint, basically. So based on so this is the little reading that it will give you. And then you can easily identify, if you have this fancy laser, you can easily identify your organism. All right, so that's everything for phenotype. The next part is genotype. So you're looking through specific nucleotide sequences. And that gets back to probes. We talked about in the last chapter about making probes. And so, but the problem with detecting specific sequences is that you can only do one sequence when you're doing this type. And they use it for quick tests. DNA first, so that you don't miss if you only have a tiny amount of DNA and you're not able to see it in your reading. of your, you're not able to see it with your little sticky things, with your, what are they called? Probes. If you're not able to see your probes, like if you're looking like, is this fluorescent or not, and you just don't have enough DNA and it is there, that would be a false negative test. Or if you're going to have, you can also have a false positive test if you have a similar organism that will pick up those, that set of genes too. So she did mention that. The other thing you can do is you can sequence ribosomal RNA or ribosomal, I put NDA, ribosomal DNA, which is a newer thing they said, but she didn't really get into detail with that about DNA versus RNA. But you are looking at the ribosomes, and the reason why you use ribosomes is because they don't change. And I kind of just remembered, like, and then when you're looking at strain differences, that's important when you're looking at certain diseases or foodborne illnesses or if you have a strain that comes back with a particular biovar, that's the result of its biochemical test. If you have a serovar, that means you have a similar serological test. Coronavirus popping up on my phone. The next thing is, let's see, I don't think she got into that. About that. So that's kind of like, I think the gel, yeah, you're going to use the gel electrophoresis. So it's like, instead of using it to like in her notes, but she shows you the gel electrophoresis. Yeah. So this is how you can identify different strains. So for example, if you see like these three, you know, a lot of these look the same. This one's different, but these ones look the same. So those would probably all be the same strain. And they have different restriction, fragment length, polymorphisms. So that's what you're using when you're doing the gel electrophoresis on my sheet. I guess it got cut off. But that just means that you were on your strain, and that's an antibiogram or an antibiotic susceptibility pattern. Back in time. You can also look at mutations because you know the mutation rate. That's how you know. That's how you can have an mutation chronometer, because you know the mutation rate. So that was when she talked about HIV back in possibly being here back in the 20s and 30s, because we can look at the difference between the strain that's in Africa and the strain that we have here. And that also gets into classification, especially when we have horizontal gene transfer, and we get into some things later on. Okay, yeah, right here. To help avoid some of those problems with molecular analysis. Like she talks about genomic islands. Let's see, is that in this one? Okay, yeah. So when you're classifying, you don't want to be classifying an organism based on a genomic island that's from a totally different kind of organism. Or RNA, so ribosomal RNA, DNA. And then the other way that you can tell how related something is if you basically melt the DNA of both and then mix them together and see how much of it comes back together. So that's DNA hybridization, which I don't know how much she talks about that. It's not okay. Similarity of nucleotide sequences. Okay, so however close they are is how much they will anneal back together. But there are some issues with that, she mentioned. And then the other thing that you can do is look at your base ratios that, I don't know if I'm sure there's other ways, but yeah, well, here you go, DNA melting curve. So you can see the organisms are not related. And if they are the same, it doesn't necessarily mean they're related, but if they're probably related, and that's just another way that you can know that they're close. Got through that one pretty quickly. And that there's rules for nomenclature is the last thing I have on my study sheet, but we got through that faster. And then here's that little sheet in the back where you've got your summary sheet in the back of the chapter. Okay, so The next thing I put out that I made for chapter 12, chapter 11, and chapter 12, it was so helpful. I wish there was a way that I could show it on the screen, some kind of way, but that's way too fancy for me. So I guess we'll just talk through. So for archaea, thermophiles, and all of that is on my little paper that I guess I didn't bring with me. Okay, so methanogens. So again, that phylum. They produce methane, and they are mesophilic and non-halophilic, so they're kind of balanced other than they make methane. Oh, the other important thing about that is that they're obligate anaerobes. So that's why when she explains, like when you walk in the swamp, you're killing them because they cannot be exposed to air, they will die. Extreme halophiles. The interesting thing about that, I mean, we should probably know like what that is and what salt concentration, but that's how they survive being in such an acidic and hot environment, is because they have that monolayer. And then hyperthermophiles are chemolithotrophic autotrophs, so they're the primary producers in their environment, which is really important, and they can be in either Crenarchaeota or Euryarchaeota. So that's everything for archaea. So she gives us 5 phyla, and she gives us organisms, and then for the biggest phyla, which is Euryarchaeota, she gives us 4 groups. So then the next thing is bacteria, and I don't have my other thing. Let me see if I can leave you and find my page. Okay, here we go. Okay, so now I can look at it. So if you have it printed out, you can look at yours. Okay, so got through Euryarchaea. Next thing is bacteria. She gave us 6 different phyla under bacteria, which I have on my page, and that's what I'm looking at. Okay, so Proteobacteria has the most organisms. They're gram-negative. They're the largest group, including more than a third of described species of bacteria are in Proteobacteria. So all of these are gram-negative. So when you look back at that list of organisms to know, everything that's on there, a lot of the stuff that's on there is going to be, let's see, everything that's gram-negative. So those things, E. coli, Salmonella, Pseudomonas. Okay. So most of these things she gave examples of in her slideshow. So she might say, I'll just read through it, and then this is really straightforward because it's just kind of Pseudomonas is honey badger. Vibrio is cholera and necrotizing fasciitis. Escherichia is E. coli. Neisseria is Shigella causes dysentery. Helicobacter causes stomach ulcers. Phylum Firmicutes, they are all gram-positive. Gas, gangrene, botulism, and C. diff. The next one was actinobacteria, include a lot of antibiotic producers, tuberculosis or leprosy, and streptomyces, which makes an antibiotic, and then actinomyces, which I don't really know what that is, but it was in her notes. It's a soil bacteria, I'm guessing. It's just a regular old soil bacteria. Maybe that was what she said is when you smell the rain. Okay, the next is phylum Cyanobacteria. Those used to be considered blue-green algae. She did not give us an example of cyanobacteria, but they use photosynthesis and they have chlorophyll. Phylum Chlamydiae, our answer is phylum Spirochaetes, and those are gram-negative. They grow in the mouth. Some are STDs. So you have treponema. The chapter we just looked at was learning how to classify, how to organize these different organisms. And then this is the organization of the organisms that she feels like paper up for that. Okay. So let me find her stuff. Okay, so I'll just overview the 5 supergroups that she gave us, and then kind of go through each one of them. But this is all just kind of straightforward from supergroup SAR, supergroup Excavata, supergroup Epistoconta. And I missed Amoebozoa, supergroup Amoebozoa. So with Archaeplastida, those are red and green algae, and they have plastids. The example she gave us was red. Don't think that red algae are considered in Kingdom Plantae, but Kingdom Plantae is found in supergroup Archaeplastida. So that's probably a question that I have. Is red algae in Kingdom Plantae? In supergroups SAR stands for Straminophilus alveolates and rosaria. Most have plastids, again, like similar to the Archaeplastida. The examples that she gave us 3 groups underneath that. So brown algae is multicellular. It likes to live in cold coastal oceans, which live in corals, and a specific species of dinoflagellate called Carina brevis, which causes red tide in the Gulf of Mexico. The group that she gave us is Apicomplexans. That's under supergroup SAR. They're non-modal obligate parasites with complex life cycles. So I kind of tried Plasmodium and Toxoplasma. So Plasmodium is malaria and Toxoplasma causes birth defects. Supergroup Excavata contains zooflagellates, which I felt like was a little confusing because you have its supergroup SAR. Confusing to me. So I don't know if I don't have this organized right or if I missed something, but that was the way that I understood it from the notes. Okay. So where I had gotten where it says zooflagellates is from the very first slide where it says that under Excavata. So maybe I just need to clarify that's correct. But the examples that she gave us for Excavata, she gave us 3 groups and gave us examples of each of those groups. So she gave us parabasalids, which are unicellular flagellated endosimians, and they don't have mitochondria. That was the interesting thing about parabasalids. And it causes, or one of the species is Trichomonas vaginalis, which is a sexually transmitted protist, considered a protist, kind of these loose definitions. Then diplomonad, so if you think of di, like di meaning 2, it has 2 nuclei. So it's unicellular, 2 flagella endosimians. So similar to the parabasalids, but it also doesn't have mitochondria, but it has 2 nuclei. And an example of a diplomonad is Giardia lamblia, which is in beavers. And then the next group she gave us was kinetoplastids, which have kinetoplasts. So that's pretty easy. You can remember what's specific about that group if you know it. So kinetoplastids are unicellular flagellated and they have kinetoplasts. Examples she gave us were Trypanosoma cruzi, which causes Chagas, and Trypanosoma brucei, which is African sleeping sickness. I wasn't really familiar with those, so I'm just going to have to learn them. And the other thing she gave us was another group. I need to make that a different color because that's a separate group. The Heterolobosians. All right, so then the next thing, I guess, what kind of cell is it? Is it a white blood cell that goes around eating everything? I haven't taken any PD, so I don't know. Eating everything. And the example that she gave of an actual amoeba is Entamoeba histolytica, which causes amoebic dysentery, and it is an amoeba. It is not amoeboid, meaning like an amoeba. It is actually an amoeba. So the next group is, I think she actually took a break in cellular or plasmodial. Cellular are single amoeba-like cells, which can turn into a slug if they need to or find themselves in, needing to kind of group together. And then plasmodial are large superamoebas, which ingest organic debris. So they look like fungi, but when they are doing molecular analysis, they're like, oh, okay, they're not fungi. Water molds form white threads onto is supergroup Epistoconta, and underneath that is kingdom fungi and kingdom animalia. Under kingdom fungi, we have yeast, molds, mushrooms, Those are heterotrophic saprotrophs, so they get their food from somewhere else, but they have to absorb rhizoids, sporangia, and conidiophores, which I don't know what those are. I need to go learn that. She gave us 1, 2, 3, 4, 5, 6 phyla under kingdom fungi, and I probably won't be able to pronounce all of these, and that causes that terrible disease in frogs, which is very, very long and difficult to probably spell and pronounce. Batrachochytrium dendrobatitis. So if you want to learn that, you should get a bonus point. So the next one is, I skipped phylum Microsporidia. Those infect hosts with spores in immunocompromised patients. Or people, I guess. And then phylum Zygomycota is responsible for black red mold. One of the groups that she gave us was Rhizopus. I think that's the genus name. And they have sporangia, which is one of those vocabulary words. And those are like types of spores or reproductive structures that they have. Phylum Glomericomycota or arbuscular mycorrhizal fungi. So those are the ones that are beneficial for plants and work in a happy relationship with Ascomycetes. Penicillium is in this group, so penicillin comes from it. Also pathogens, morels, truffles, and lichens, and ergo of rye. So some of those diseases, things you can get are Aspergillus and Candida. And an example that she gave us was the white nose syndrome in bats of Geomyces destructans. The last phylum under kingdom fungi, under Episthocanta supergroup, is Basidiomycota, and that one is club fungi and is responsible for lots of deaths. Amanita is responsible for 90% of mushroom-related deaths. And then at the bottom of my paper, I have that lichens is a likely kind of like, take some stuff away from it. It's not really a beneficial relationship. Okay, so kingdom Animalia, that's all the stuff. We're going to get into worms and more worms and arthropods. So I was very itchy that day when she was teaching. Okay, so the first one, she gave us phylum Platyhelminthes, which I don't know how to pronounce this. A coelomate means that they don't have, I'm far away in my book from this now, that they don't have a body cavity, or they have something about their body cavity. Okay, so yeah, they don't have a body cavity, so they're a coelomate. They have flame cells, so they have a primitive excretory system, or have an excretory system, I guess. Yes, have an excretory system. They don't have cephalization, so not really like a lot of a brain thing going on. And there were 2 classes underneath phylum Platyhelminthes, class Cestota and class Trematota. So under Cestota, you have tapeworms, which have a scolex. They have segments. They have proglottid. That's the segments. The segments, but they're segmented worms. They their segments are called proglottids. They don't have a digestive system. Checking the message over here. Okay, and then under class Trematota, so the class is underneath phylum Platyhelminthes, is flukes Platyhelminthes, including class Cestota and class Trematota. Then we have phylum Nematota, which is roundworms, which are colorless, non-segmented, and they have they're a pseudocoelom. I don't know what that means. Pseudocoelom. Okay, so that's a false body cavity, incompletely lined by mesoderm. Okay. And the examples that she gave us were Ascaris, Lumbricoides, which causes ascaris, and then Wuchereria bancrofti, which causes elephantiasis. Okay, so that goes under phylum. It could be through mechanical or being a reservoir. And then, so she didn't give us like anything underneath that, but she did name some organisms that were in it. So mosquitoes, fleas, lice, and ticks. So mosquitoes transmit malaria. Lice are wingless insects, and body lice transmit bacterial diseases, but head lice don't. And then ticks are arachnids. They transmit Rocky Mountain spotted fever, Lyme disease. Oh, and mites. Mites are tiny arachnids, which can cause asthma, chiggers, and scabies. Okay. So, I did not. Do you need something, Autumn? Do you need something?
I might be sick because I'm hungry and.
You might be sick because you're hungry.
No, because Grace touched me and she said I'm hungry.
Well, if you're extra cold, I don't think that means that you're sick.
And I'm hungry and my throat hurts.
You're hungry and your throat hurts.
And normally Miss Kim lets me get some food.
Let me see out now. I have a sugar cookie. Why don't you eat a sugar cookie and see if it helps your sickness go away? Okay, so I don't know if I want to try and go through chapter 13 because I have not gotten to study that one very well. So maybe I'll have to study that one. I have not made my little paper for that either. So if nobody else has any questions, Felicia doesn't have any questions because she's the only one here. Then I guess that's pretty much it. I might be able to spend some time tomorrow. Let's see. Let me log into Canvas from here because my phone's not wanting to work and see what those comments are. Okay, so I might be able to spend some time tomorrow if anybody would like to interact about some of these things. I basically just kind of talked through her slideshows up to chapter 13 in like an hour and a half. So if that helps you review, I'm glad. Let's see. Sorry, I'm trying to find my discussions over here. Okay. Yay. Okay. All right. It doesn't look like there's anything new on the discussions. So I guess this is it. If anybody else has any other suggestions on how to study or have any specific things, all I can say is review the Quizlets. The most helpful thing for me is listening to her lectures, and then it's nice because we can skip through some of the discussions if you're trying to just remember the content. So I'll do that while I'm driving and just listen through it. And then just studying the Quizlet. So between that and then my organizational charts, which I'll go back and fix some of these little mistakes that I ran across while we were reading. Or if anybody else has comments or questions or anything like that, feel free to pass it on. And I think... What was you before you found the video? Oh, I didn't see when you sent that, so I don't remember. I can't imagine that would be interesting for anybody else, but anyway, so will we manage to make it through all those chapters in an hour and a half. So that's actually pretty good. So yay, Felicia. Thank you for being here for me. So I didn't feel like I was just talking to myself. And I guess I'll go ahead and end the stream. And I think it should be able to, like, people should be able to go back and watch it again or skip through it if they're looking for a specific part. But let me know if anybody has any other suggestions for studying or, working through the material. And good luck to everybody.